Okay, so today we're going to continue on with our discussion of the reproductive system. And we're going to spend most of the time today talking about gametes and their development. So the development of our fully mature sperm and egg cells, I think will be mostly what our focus for today will be. So when we left off last time, we had been talking about the structures of the male reproductive system, and we had talked about how we get set up with different anatomy, a different sort of base plan for male versus female anatomy. So we'll touch on both today, but when we left off, we were talking about the sperm. So we know the sperm are produced in the male gonads, the testes. In these structures, the seminiferous tubules that have these different types of cells, Sertoli cells lining the lumen of the tubules, and then the Leydig cells producing testosterone kind of scattered between. Okay. So our goal is to produce fully mature motile sperm, so sperm that can move. So here's what that looks like. So we're going to have three basic parts to a sperm. So we're going to have a head here at the top. There's going to be a midpiece, and there's going to be a tail. So we're trying to swim the head forward, and we're using energy being produced by that midpiece to create whip-like movements of the tail to get that to happen. Okay. So the head is really the capsule with all the information in that gamete. So all the information we're trying to carry forward is located in the head. So we're going to look at two separate parts of it, kind of. So the nucleus here is within the head. Okay, so we have that nucleus. So this is where we're going to contain all those chromosomes. So our actual genetic information. Sperm will be haploid, so half the genetic information as usual on its way to fertilization. Okay, so that's in the nucleus. But we have this other little compartment here called the acrosome. So you see this other bit here at the point. Okay. So the acrosome is going to contain a bunch of enzymes that are necessary for fertilization. So just like a lot of these other processes in our body, we're going to need to do modification. We're going to need to break stuff down. Specifically, when we're thinking about sperm and fertilization, we're going to need to break into an egg cell. Okay, so some of the enzymes necessary for fertilization are going to be located kind of in this pocket in the head of the sperm. Okay. In the midpiece, you'll notice all these red dots. Those red dots are there to represent the mitochondria. Mitochondria are super important. Their main role, right, is the powerhouse of the cell, aka they're helping you produce ATP. Right, so we have lots of mitochondria in this midpiece of the sperm, so that we can do a lot of cellular respiration, produce a lot of ATP, because we're going to be swimming. We're going to be moving that tail with these whip-like movements. We need energy for that. So those mitochondria are there to produce that energy. So that's what's in the midpiece. And then we have the tail, or the flagella, right, is the type of structure this is. So this whip-like tail is going to move back and forth, propel this sperm forward. So how do we get there? So what we're looking at now is an even more limited view of these cross sections we were looking at through the testes on Wednesday. So we had before, right, like a testis. I'm going to draw the epididymis on top, right? We saw that there were seminiferous tubules throughout, right? And we took a little cutaway before to look at a cross section. Right, so we cut like that. And then we saw that we had essentially like a little circle of cells surrounding a lumen in the center, right? So those were our Sertoli cells. And then we had Leydig cells over here, and the lumen was here. I'm just orienting us, right? So what we're looking at now is just like this segment, right? So we're looking at two of those neighboring cells, the space between them. And then so the outside here, this basement membrane is facing like this outside wall of the seminiferous tubule. And here we see the lumen, so like the hole through the center of that tubule on the inside. So what we're tracking here across the wall of the seminiferous tubule is sperm development starting out near the outside part of the wall of this tubule, near the basement membrane. And the sperm are being produced and maturing as we move into the center towards the lumen. So that's why we're zooming in here. So we mentioned before that those Sertoli cells have tight junctions, so forming a seal around this lumen. So that's also a factor as we think about the stages of development of spermatogonia into spermatozoa eventually, is that we really can form sort of protected environments on either side of that tight junction. So when your book refers to there being like a basement compartment over here versus a luminal compartment over here, that's possible because we have these tight junctions. However, they are going to open up periodically to allow these developing sperm to move down and towards the lumen. Okay. So the process of producing sperm is called spermatogenesis. So genesis for creation, spermato for the sperm. Right. So we start out with our germline cells, our spermatogonia, out on the edges here. So we have spermatogonia up here by the basement membrane, out near the edges of these seminiferous tubules. So the spermatogonia or germline cells, our progenitor cells, are what we start out with. Okay. So we're starting out with them. We are going to be taking these spermatogonia, running them through meiosis, ending up with our gametes, our spermatozoa, which will mature eventually into fully mature sperm as they exit the testes and go through the epididymis. Okay. But it's important to realize that one of the reasons fertility is maintained throughout the lifespan in males is that these spermatogonia replicate themselves and essentially in that way regenerate, right? So we have these spermatogonia up here at the edge, and we are going to be taking some of them and dividing them, right? But the rest of them are undergoing mitosis over and over again. So we're making more and more copies. That's what this dotted line is for. So it's this regenerative process, this mitosis happening with the spermatogonia that is going to retain fertility in the male reproductive system. This is going to be a contrast to when we talk about the female reproductive system, because our analogous cells don't undergo mitosis in this way in the female reproductive system. So this is a difference, an important one. Okay. So we start out with our spermatogonia. Okay. When we're ready to start making some sperm cells, which we're doing all the time pretty much. So we're going to start going into meiosis. So we do name the different stages essentially of development of these cells. So we're setting ourselves up for meiosis here, creating our primary spermatocytes as we duplicate all our chromosomes and get ready to divide. So that's what we start out with here. So we're traveling down the spermatogonia, we're just themselves making more and more and more. When we start to differentiate, we're duplicating, setting up for meiosis, becoming primary spermatocytes. You can see we're starting to move down between the Sertoli cells at this point. So from one spermatogonia, so from one cell here, you'd end up with two primary spermatocytes with a diploid amount of information. So we still have all our genetic information here. So that's what this 2N is, and 2 telling us that this has happened twice. Okay. Then we're going to undergo meiosis. Meiosis has 2 phases, meiosis 1 and meiosis 2. So hopefully you recall this somewhat from your prerequisite courses, but basically we're duplicating all our information, we're having it, and then we're having it again. And this is a difference between meiosis and mitosis. Mitosis, we just duplicated the information, cut it in half, ended up with 2 cells that were identical to the parent cell. But in meiosis, because we duplicate, pull in half, then pull in half again, we end up with only half the information. We end up with haploid cells, and those are our gametes, in this case, our sperm cells that we end up with at the end. Okay. So when we undergo meiosis 1, what we produce at the end of this first phase of meiosis are the secondary spermatocytes. Okay, so at this point already, we've pulled apart sets of chromosomes. So we are now haploid cells. So we have half the information, but all that information within each cell is still duplicated in these secondary spermatocytes. And then meiosis 2 completes the meiotic process. So we're completing meiosis, ending up with our spermatitids, which would be the 4 cells, each of which is haploid. So each of them includes like just one of each chromosome, just one copy of the information. And each of these spermatitids are different from each other and different from the original parent cells. Okay. So this process, this whole thing, is just showing you what our specific names are with regards to the male reproductive system for cells that you would have been tracing through in your earlier biology courses, right? This is just a description of what's going on in meiosis, okay? We just have specific terms if we're talking about male human reproductive anatomy. As we're moving forward though, we're going to continue to modify those cells, and what we put down into the lumen are the spermatozoa. So we can see that we have gone further than just sort of dividing up our genetic information and mixing it around a little bit. We've also, through signaling from our hormones, from our gonadotropins, we've started to change the structure of that cell as well, right? So we can see we start to have that structure with the head, the midpiece, and the tail as we are going into the lumen. Okay, so those are the spermatozoa. So spermatitids, when we just have a sort of basic looking cell, we've divided the genetic information with meiosis. Spermatozoa, they're kind of taking shape and we're putting them into the lumen. Okay. However, they're actually not ready yet to actually go fertilize anything. The spermatozoa that are getting released into the lumen of the seminiferous tubules are still immotile. So think immobile, they can't swim yet, basically is what we're saying. So they have some of those structures, they're differentiated a little bit, but they're not ready to do their job, basically. So they're gonna hang around for actually about like 20 days before they develop enough to gain that ability to move. Okay. So the spermatogenesis was what we were tracing through in that image on the previous slide. So it was happening from the basement membrane that was out at the edges of that tubule, basically, right? Towards the lumen, right? We had mitosis happening to regenerate more spermatogonia out on the edges, while another spermatogonium is undergoing meiosis, moving inwards, eventually producing those spermatozoa that are hanging out in the lumen, but they're not ready to move. In order to get those spermatozoa to be fully mature sperm that have motility, that are able to move, it's actually going to take until we move them into the epididymis. Okay, so we're moving from the, here's the testis, right? There's our epididymis kind of sitting on top, tail going up into the vas deferens. Okay. Right. So our seminiferous tubules were here. So this is where we had our spermatozoa. Right? They move in to the epididymis by peristalsis, and also by the fact that there's fluid flowing in that lumen of the seminiferous tubules. Right? So there's movement going on, and that continual flow of fluid is also kind of pushing them forward. Okay? So then they're in the epididymis, and this is where they undergo some more modification, right? There are enzymes involved. I don't know the exact details, but this is where they acquire motility, is when they're in the epididymis, moving towards the vas deferens. So they're acquiring motility here in the epididymis, and then they're going to go hang out in the beginning parts of the vas deferens. Okay, so they get there again by peristalsis, just this kind of segmental movement. And they're going to stay there in the vas deferens until ejaculation. Okay, so this is where they're hanging out until they're ready to go somewhere. So to complete our understanding of all the processes going up to fertilization, we then want to know about the sexual response in males. We want to know about how ejaculation happens and why. Okay. So the whole sexual response is controlled by the autonomic nervous system. So remember, that's our sympathetics, our parasympathetics, and it's happening relatively undercover, right? Like not totally conscious control, right? It's automatic. Okay. So there are going to be a number of spinal reflexes involved here in the sexual response. And there's spinal reflexes involving the autonomic nervous system, so parasympathetics and sympathetics. When we think about the sexual response in males, we'll think about, I tend to think more about two phases, but we'll name all three. Think of the middle one as kind of a transition point. Okay. So first we're going to think about erection. So we're thinking about creating, moving from a flaccid penis to a one that's erect, standing upright, swollen with blood. In emission, think about all our glands adding in their fluids. So fluids coming from the prostate gland, from those seminal vesicles, from the bulbourethral glands. We're going to see a switch in the part of the nervous system that's responsible. Okay. And then in ejaculation, we're thinking about the semen actually exiting the body through the penis. And that's under the control of the sympathetic nervous system. Okay. So all of this is going to start out with the parasympathetic nervous system. This is what causes erection, okay? And we'll walk through a flowchart of how this is happening. But basically, parasympathetics, that rest and digest part, is going to cause more blood flow to the penis, and it's going to cause less venous return from the erectile bodies in that penis. So the blood gets stuck there, and that press of fluid against the walls of those erectile tissues causes the penis to stand upright. Then we're going to see a switch from the parasympathetic nervous system to sympathetic, so to the other half of our autonomic nervous system. So this is when during emission, those glands are going to start emitting their fluids. And then during ejaculation, we'll project that semen forward, also under control of the sympathetic nervous system. A slightly off-color mnemonic that people use to remember this is point and shoot, parasympathetic versus sympathetic for erection and ejaculation. So if you want a mnemonic to remember which part of the nervous system we're talking about. So the stimuli for going through these phases are multifaceted. So we have lots of potential stimuli. Primary ones, we're going to be thinking about mechanoreceptors in the penis, so touch sensation, essentially, right? As well as mechanoreceptors in other erogenous zones. And this is going to be partially because we have linkage, right, between all these mechanoreceptors out in our body and higher brain centers, right? So there's this complicated network of nerves and thoughts and integration going on. So we're going to have descending signals coming back down from the higher brain centers, putting together all this information, as well as data input from the mechanoreceptors locally themselves. So if we want to look at this in flowchart form, we're going to be starting out with mechanical stimulation of receptors, especially in the penis, so actual touch sensation, as well as neural pathways in the brain sending their signals down and out. Okay, so thoughts, information from other regions of the body, all getting kind of mixed together, interpreted by the brain, sending input out to the rest of the body. Okay, so both of these types of information are going into our spinal cord. Okay. So we're going into the spinal cord. Okay. And here's where we see that autonomic control. Okay, so we're going to see that what's happening is that we are decreasing our activity in the sympathetic neurons and increasing activity in parasympathetic neurons to the penis, okay, as a response to these initial stimuli. Okay, so parasympathetics are under control here. We're kind of turning off the sympathetic nervous system at first. Okay. And It's kind of special that the parasympathetic neurons in the penis themselves are going to be changing the smooth muscle of arterioles in the penis. Usually we saw that that was to do with sympathetics in other arterioles in the body, but in the penis, we have parasympathetic innervation to those arterioles. And what we're going to see is relaxation of smooth muscle. Okay, so specifically in the arterioles, in the erectile tissues in the penis. So we're thinking about a blood vessel, right? So if we relax smooth muscle, we go from a little small tube, small blood vessel, to a bigger blood vessel as we relax that muscle. So what we've done is increase blood flow, right? So we increase blood flow through this parasympathetic innervation, okay? Which means that we now have a bunch of blood volume and blood pressure rushing into the penis. Okay. And those arterioles and arteries and little bits of capillaries, they're sitting right next to veins, right? Actually, they're sitting next to a plexus of veins, right? But as they get really big and full of blood, they're kind of pushing on the walls of those veins. You can kind of think of it like, right? So they're going to compress those veins as they're expanding in this high volume, high pressure situation. They're going to kind of close those veins over just by pushing in from the sides, okay? So in addition to having a bunch of blood flowing in, because these veins are getting compressed, we're not going to be able to have that blood to flow out anymore. And this is all a big positive feedback loop, producing erection, okay? So these two things together, the fact that we have more blood flow and we are preventing blood from leaving the penis, right? You have more blood flow, begins the process of erection, compress the veins, continued blood flow, positive feedback loop, creating greater and greater and greater erection. So that's what this is a kind of a map of, right? So we think about those initial parts of the response kind of here in the middle, getting us to erection. So once we establish erection, so an erect penis, straight, full of blood, mechanical stimulation continuing is going to cause a switch from this erection phase to emission and then moving forward to ejaculation. So the mechanical stimulation continuing is just going to kind of set us on a pathway for the neural activity to switch from parasympathetic to sympathetic control. So it's just kind of part of this timeline, okay? So we're switching over to sympathetic control. And at this point, the epididymis is going to start to contract, as is the vas deferens and the ejaculatory duct. So we're moving those spermatozoa that have been hanging out in the epididymis, gaining motility and therefore becoming like mature sperm. Maybe they're hanging out in the first parts of the vas deferens, right? They're all getting pushed forward towards fluids in the seminal vesicle, forming that ejaculatory duct. Prostate is giving off its fluids. All of this stuff is moving forward into the urethra, okay? So we're just kind of moving everything along down the pipes in this emission phase, including now these various secretions from our glands. As the response to mechanical stimulation continues, we are now moving more fully into sympathetic activity, right? We switched from parasympathetic to sympathetics, causing emission, causing these glands to secrete their fluids. And now during ejaculation, we really just have a really strong sympathetic signal, okay? So this is going to stimulate contractions of smooth muscle in the urethra now, right? So in the urethra, so that tube exiting out the penis, as well as in skeletal muscle at the base of the penis, also stimulating contractions there, okay? And as they pulse, essentially, they're going to move semen through the urethra and out of the body. At the same time, as we're contracting these muscles, this is also stimulating closure of the urethral sphincter, internal one. And this is important because that urethra is also connected up to the bladder, right? So as we close that sphincter, we're cutting off that connection to the bladder. So we're ending up with semen full of sperm and these enzymes and fluids from our glands coming out instead of mixing in with urine. So that is our understanding of the male reproductive system. Now we're going to move briefly into the first part of the female reproductive system. Okay. So when we're thinking about the female reproductive system, we're going to see some similar structures, some analogous structures, I guess I'll say. Cell types, we'll see similar things going on, but we're going to be contrasting a number of things that are different. One of the things that is different is that we're going to have cyclical changes in activity within the female reproductive system, called the menstrual cycle. So we're going to have cyclical changes in hormones, in what cells are developing, how and why, where they are, what phase they're in. The timing of events is different than in the male system, which produces some really important results in the body. It's also true that this means we are going to end up with restricted periods of fertility in the female reproductive system. So specifically, ova are only available to be fertilized at certain points within the menstrual cycle. So the cells that we need for fertilization are only released during ovulation, which occurs roughly halfway through the menstrual cycle. We'll talk about exact numbers as we move forward. All right, and they're only there very briefly, and then they're gone. Okay. Another difference is that we have a limited gamete pool, essentially. So in males, those spermatogonia, we have one of them just duplicating over and over forever and forever, right? But in females, we're actually going to see that the pool of gametes is established at birth. So the timing of actually when these phases of meiosis are happening is different, partially resulting in this. So we'll take a look at how that works. Okay. So we'll take a little look at each of these structures real quick. Okay, so when we're talking female reproductive system, right, we're talking about ovaries in the pelvis, right, hanging out, ready for the cells that they're producing and releasing to be caught by the uterine tubes or fallopian tubes. I got that there. All right, so that's the structure here, hanging around. Our cells would then travel into the uterus. Okay, so here we see the uterus. The bottom third or so of the uterus is another structure called the cervix, right? Sometimes we'll refer to it separately, often we'll refer to it separately. Okay, which projects into the vaginal canal, which leads out of the body. Okay, so that's the tube we are talking about. And we can already see that this is one difference here, right? Is when we were tracing through the male reproductive tract, we saw that we linked up with the urethra and we kind of shared a set of tubes there. Not happening in our female reproductive system, right? We have our whole own separate pathway. The urinary system is separate, bladder, urethra, separate opening anterior to these structures. Okay. And now we're looking kind of within one ovary. So we're going to cut open an ovary and look at it as we start to think about what the cells are doing. Okay. All right. So what's happening when we talk about the female reproductive system and the menstrual cycle is we are going to see essentially we're selecting little follicles, so little cells that already exist in the ovary. They're starting to develop. One will become dominant, become an egg cell that gets released, essentially. All the others die off. And then this process happens over and over again every month. So part of the reason that female fertility is kind of started at birth is that these follicles are already there in the ovary. We're not making them new. We're not doing mitosis over and over again. We are starting out with these primordial follicles here in the ovary set to go. Okay. So here we see those primordial follicles. As they develop, we're going to see that they get bigger, and we're going to name parts of them differently, as well as the cells surrounding them. So we can see the primordial follicle here. We can see that there's an oocyte within it, as well as some follicle cells forming kind of like a shell. We can see that as it gets bigger, we start to name granulosa cells. And now we're just going to call them follicles as they develop. We can then see that we're going to form another sort of development around the collection of cells at this point called our theca cells. And then our follicle here is called a Graafian follicle. So just like when we were going through spermatogenesis, right, we had names for different phases, right? These cells are essentially going to take on different names during different stages of development. But here we're actually stretching out development in a different way. Okay. So we're beginning with our primordial follicle, which has a little oocyte within it. We have these follicle cells around it, right? And then we are going to start to have these granulosa cells kind of around. Okay, so the granulosa cells are going to be targets for hormones. So they're going to be target cells for estrogen and for FSH, our follicle stimulating hormone. Yeah. In response, they're going to secrete chemical messengers, okay, including we're going to be secreting inhibin from granulosa cells. This is going to provide a negative feedback loop back towards FSH, our follicle stimulating hormone, which when we link together all the different pieces of what's going on, is going to be an important part of hormonal switches we kind of see during the menstrual cycle. Yeah. Another thing that is happening is that we are able to transport nutrients to the oocytes so that egg cell inside through gap junctions. And we are also secreting estrogens in response to in addition to being target cells of estrogen. So here's a picture that looks more similar to what was going on in the male reproductive system, but note that we're now not looking like at a specific physical place, right? This is up and down is not like our Sertolia cell top and bottom anymore. What we're looking at is different time periods, right? So when we're talking about, okay, so when would mitosis happen? When would you get more oogonia? So those are the germline cells analogous to our spermatogonia. This is actually happening during fetal life, okay? So this is earlier in development when we would be producing our total pool, essentially, of ultimate egg cells, okay? So during oogenesis, we're actually starting during fetal life, right? So we have kind of a time scale here instead of a like physical, where are we scale, right? So mitosis and differentiation producing more oogonia happens during fetal life, producing primary oocytes. So we're like set up for meiosis. And we're going to pause here, kind of part way into meiosis at birth, essentially. So we're going to be in this phase of mitotic arrest. Okay. So in this overall process, we're trying to go from oogonia to mature ova. Ovum is singular. Those are like our egg cells, the fully mature ones, right? But we're setting up even before birth, right? So we're starting this process, getting ready for meiosis and pausing before birth, meaning that our total number of oogonia is fixed, right? Because we've sort of set everything in motion and then just paused, right? So we can't go back and do more mitosis, essentially. Okay. So that meiosis starts in fetal life, pauses, and then continues during the menstrual phase. And actually, meiosis isn't fully complete until after fertilization, right? So it's a really kind of complicated timeline we have going on here, right? Spanning many, many years, decades, potentially, right? Hopefully decades, actually, right? So we're starting in fetal life, right? Meiosis 1 only completes kind of just prior to ovulation. So it's going to happen on essentially a single or a couple developing ova at any one time. It's not going to happen to all of them at once, right? And then we don't undergo meiosis 2 until after fertilization. So this would be after intercourse, after a sperm has penetrated the ova, right? Only at that point are we actually going to kind of trigger this second part of meiosis. So this is a more complicated process in many ways. So another piece of the complication, in addition to the fact that fully undergoing meiosis and getting to our gametes is a really long time scale, is that we have the menstrual cycle cycling on roughly a monthly-ish basis. And we have different phases of what's going on in the ovary and the uterus. We have cycling happening in the ovary and in the uterus, changes in parallel in response to hormones coming down from the hypothalamus and kind of back signaling. So we're going to have to be mapping a lot of things kind of together to fully understand the female reproductive system. So what I do want us to introduce today briefly is we're just going to briefly think about the ovarian cycle. So we're first going to start by thinking about what's happening in the ovaries, which we're going to be able to break down into two phases, a follicular phase and the luteal phase. And then next week, we'll be pairing that up with what's going on in the uterus. So uterine changes in the uterine cycle to fully understand the menstrual cycle before we then move forward and think about fertilization, pregnancy, and some more of the hormone stuff going on. Yeah. Overall, the events in all parts of this are going to be caused by and then influencing changes in estrogen and progesterone concentrations. So hormones are really strongly involved here. Yeah. So we're going to start out with the ovarian cycle. Okay. So the overall menstrual cycle, this is going to be true when we think ovarian cycle or uterine cycle, because they are going to pair up, runs roughly 28 days. Okay, so that's why I said monthly-ish months. Most of them are not 28 days, except sometimes February, right? But your average standard ovarian cycle is. Okay, so we're going to split it in half, essentially. And the halfway point is ovulation. Okay, so ovulation is going to be happening around day 14. Okay, so might be surprising to realize that we actually start counting with the first day of menstruation. So the first day we would notice signs of blood and shedding of the uterine lining, right? That's the beginning of our phase. Then we have roughly 2 weeks until ovulation, when we're going to see the release of our egg cell, essentially. Okay, so that's happening at ovulation. And you can see, even before we go through naming what's going on in here, is that what's going on roughly during this follicular phase is we're having the growth of those primordial follicles. They're getting bigger and bigger. We're developing different structures, right? We'll be producing different hormones until that cell is released. During the follicular phase, we're developing a follicle, okay? And then during the second half, so after ovulation, during the luteal phase, what we're seeing is essentially degeneration of those structures we've built up. So we released our egg cell and the remaining structures degenerate and become actually kind of a gland that starts to fade away until menstruation begins, and then we loop back to the beginning, right? So we can see this remaining structure slowly disappearing. So we're splitting the ovarian cycle in half between follicular phase and luteal phase. The halfway point is ovulation, occurring around day 14. Right? Menstruation is actually at the beginning. Okay. So thinking about that follicular phase, so the first half of this cycle. Okay. So the first half of this cycle is characterized by the fact that we have that primordial follicle, right, growing and developing. So it's going to be getting bigger. Okay? So that cell inside that's eventually going to become our mature ovum, right? This egg cell, right? Our oocyte, oo for egg, site for cell. It's growing, so we can see it getting bigger. Okay? The cells surrounding are also proliferating. So we're making more of the granulosa cells, right? So those cells kind of surrounding, okay? Actually kind of this area, okay? And we are going to have more structures forming. So we're also forming something called the zona pellucida, which is just another kind of circular area. Critically, I'd say the antrum is forming, okay? I like the antrum here. So the antrum is this thing that looks like kind of a pocket of water, essentially, and that is almost going to be its role, okay? Oop. Well, just mine turned off. Okay, cool. So what's happening here? I want to point out that we're looking at just one follicle when we look at tracing through an ovarian cycle here, but we actually begin by taking like a handful of follicles and developing them. So if we're zooming out, right, and we're thinking about an ovary, right, here's an ovary. We have some number of those primordial follicles. They're in meiotic arrest, right? And some collection of them are going to start during any menstrual cycle to start to do what we're seeing here in the follicular phase. So they're going to start to grow. So let's say I pick, I don't know, like 10 of them or something, right? Actually, I'm not going to circle 10, right? But some random collection of them are going to start to grow, okay? So it's kind of an arms race at the beginning. So it's not all of them growing, it's just some of them. But eventually, the one that's farthest ahead in development, the one that's fastest to grow, is going to take over. And the other ones that had started developing are going to just like kind of die off, okay? So when we refer to a dominant follicle, that's what it's talking about, okay? So as we go through this follicular phase, we have multiple different follicles that are kind of trying to be the winner of this race. As soon as one gets big enough, its hormonal signaling kind of takes over and the other follicles regress and die away. Okay. After this point, the corona radiata develops. So that's this kind of star-shaped stuff around the egg cell. Okay. And we have a mature follicle, which we're now just going to call a Graafian follicle. We're going to have ovulation occur. So ovulation, what's happening is that this sack of water looking thing, you can kind of think of it as a sack of water, actually bursts and therefore expels the cell on the inside. So that growing oocyte, right, within this mature follicle, this Graafian follicle, bursts out of the ovary, essentially, okay, as the antrum kind of explodes. Okay. This is under the control of hormones. Okay, so some of these hormones, you might want to look back, way back at some of our charts from the endocrine system. So we didn't focus on these ones specifically at that time, right? But like our anterior hypothalamic hormones are going to be involved here as well. So what's happening in terms of hormones is GnRH, gonadotropin releasing hormone, during the follicular phase is going to surge. So that's coming up from our hypothalamus, right? And its surge causes surge of a hormone called LH, or luteinizing hormone, okay? And this is what is eventually going to trigger that ovulation, right? So we're developing this follicle, right? It's growing, growing, growing. That LH surge triggers this kind of explosion and expulsion of that Graafian follicle, essentially that maturing oocyte, and triggers the development of this gland that we're forming from all the tissue that's left behind. So this gland is called the corpus luteum, okay, during the luteal phase. That's why it's called the luteal phase, corpus luteum. So we will later be looking at maps of these hormones and how they're mapping on and kind of triggering each other as well. So during the luteal phase, this follicle that's ruptured is becoming a gland called the corpus luteum, which is secreting estrogen and progesterone, okay? But it's also getting smaller as it dies away, okay? its maximum amount of activity, and by activity, we mean like hormone production, happens in about 10 days. And then it's degenerating in this process called luteolysis. Okay. So it's decreasing over the course of the luteal phase, decreasing production of these estrogens and progesterone. And then your body's response to that, these decreasing levels then, is going to be menstruation and the total end of this luteal phase. However, if that oocyte that was expelled, right, this oocyte, it left the ovary, it went into the uterine tube, right? If it gets fertilized in the female reproductive tract, degeneration of this doesn't happen because there's a separate kind of hormone signal that tells it to stick around. Right, so we have a lot of sort of fiddling that can go on here depending on what's happening in the body. Okay, so we're going to leave that here. We'll recap this tomorrow because I know that's a, you know, big mess or a lot to get your mind around. And we'll kick off with this next time. Have a great weekend, everybody.